All right, let's talk about the scientific method, hypotheses, and confounding variables. First off, what separates science from non-science? Well, one thing is clearly defined terminology, so we all know what we're talking about. Secondly, of course, quantifiability. Third, controlled conditions. Fourth, repeatability. And last, prediction and testability. So we're going to talk about some of these things as we go along. There's also distinction within the sciences. We often hear the terms hard versus soft sciences. Hard sciences are often referred to as sciences like biology, chemistry, and physics. Soft sciences are things like social sciences, sociology, psychology, and sometimes ecological sciences can be put into that category as well. But if you think about it, the terms hard science and soft science really pertain to all disciplines. And in many respects, all disciplines use both hard and soft approaches. Hard science generally refers to rigorous experimental approaches, whereas soft science refers to correlation, natural experiments, comparative studies, where there's not a rigorous control of the experimental units and the treatments. Hypotheses are tentative explanations of a phenomena in the natural world, and there's three key features. First is testability. It's possible to go out to collect information to actually test the predictions. Secondly, falsifiability. It would be possible by collecting those observations to actually falsify the hypothesis. And third is repeatability, in that it's defined so that others can actually go out and repeat the experiment. Experiments are usually designed to test the predictions of a hypothesis. Sometimes you may actually look at the assumptions. And there's two different ways to formulate a scientific hypothesis. The first is to describe it as a null hypothesis, and the second is as a alternative hypothesis. Scientific hypothesis can be distinguished from a statistical hypothesis. With a statistical hypothesis, you're making a statement about one or more population parameters. Hypothesis testing refers to the formal statistical procedures used to accept or reject statistical hypotheses. So an example of a scientific hypothesis is that the rate of extinction is higher on small than on large islands. The statistical hypothesis underlying this would be the average rate of extinction on a random collection of small islands is higher than the rate of extinction on a random collection of large islands. So with the statistical hypothesis, you're getting much more specific about exactly how you go about testing that prediction. So how do we translate our statistical hypothesis into an actual statistical test? Well, in our case, our null hypothesis is that the extinction rate of species on small islands, say less than 100 hectares, is equal to that of the extinction rate on large islands. And we might use a t-test for this comparison. In this equation, the subscripts L and S refer to large and small islands, respectively, and S squared with its appropriate subscript refers to the estimate of the standard deviation within each one of these groups. In the case of the t-test, we're looking at the difference in the mean effects of the treatments, in this case large and small islands, compared to the experimental error. The numerator represents the treatment difference that we're interested in, and the denominator is the within group variation, or the experimental error. And you see here that the numerator is what Ken Burnham referred to as effect size in his lecture. And it's often effect size that we're interested in. If there's something that's causing a big difference between two groups, then that's something we want to investigate. Now, it's possible in some cases there's so much variation in the system that it's hard to detect that. But if there is a big effect, then we might try to design ways to reduce that experimental error so we can really get at whether or not this effect that we're seeing is real. And of course, the statistical hypothesis then relates to the significance tests that you use. And this brings up the issue of type 1 and type 2 errors, something that you probably learned in your beginning stats course, but let's just review this. A type 1 error is a situation where your null hypothesis is true. So if you're looking at the effects of some drug treatment on blood pressure, there actually is no effect of the drug on blood pressure, but you reject that null hypothesis. In other words, you conclude that there is in fact an effect. The differences between your two groups is large enough that you conclude there is in fact a difference. In that case, you are making a type 1 error, and that is determined by the alpha level that you set for your statistical test. The other kind of test is a situation where you fail to reject your null hypothesis when in fact it is false. So in other words, there is an effect of the drug. You do your experiment, run your statistical analyses, and lo and behold, you find there's no difference in blood pressure between your two groups. That is determined by beta, and we'll get into exactly how we determine alpha and beta in a little bit. So let's talk about factors influencing type 1 and type 2 errors. Remember, type 1 error is concluding that there's an effect when in fact there is not. And the probability of this occurring is completely determined by alpha. We generally use an alpha of 0.05, but that value is not written in stone. So for example, if you're working on an endangered species, you oftentimes want to err on the side of caution. You might want to use an alpha of 0.1. So for example, when evaluating the efficiency of a new diet formula on the growth rate or survival of black-footed ferret kits, you might want to just go ahead and use that new diet, even if it costs a little more, and even if the effect is small or non-existent, because this is so important to ensure that you have healthy numbers of black-footed ferret kits produced. On the other hand, if the consequences of implementing a policy based on some statistical test are consequential and perhaps negative, you might want to set an alpha level of 0.01. So for example, if you're working for the Forest Service and you want to protect the trees around a campground so that there's shade where people are camping, you want to protect it from beetle kill, and say you find out that it'll cost $100,000 to spray those trees to hopefully protect them from the beetles. You might want to make sure that that money is really well spent, because if you spend that amount of money, that means that you can't replace somebody when they retire next month. And you really want to make sure that this treatment is going to do the trick. Another thing to be very aware of, and this will come up later when we talk about some examples, is that when you set the type 1 error rate, remember that that means that there's going to be a certain chance that you're going to make an incorrect conclusion. So for instance, if we set the alpha at 0.05, that means there's a 1 in 20 chance that you can conclude there's a difference when in fact there's not. And oftentimes what we do in the fisheries and wildlife area is we run lots and lots of statistical tests. So we go and measure 40 different habitat variables on places where we see spotted owls and places where we don't see spotted owls. And then we run statistical tests on all of those to ask, is there more down wood where we find them? Is there more tree cover? Is there taller trees, different proportions of Douglas fir or whatever it might be? And lo and behold, you come up with a few things that are different between places where they occur and where they don't occur. Well, remember, when you're doing lots of tests like this, it's almost certain that you're going to find some differences that are significant. And so what you can do in that case is you can adjust your alpha level downward, and there are actually formulas that you can use based on the number of statistical tests that you're running. Or you can just a priori say, well, I'm doing a lot of tests, so I'm only going to consider differences of 0.01 or 0.001, because you really want to know, the ones that you're certain are different between these two situations. Something to be aware of definitely in wildlife and fisheries. Moving on to type 2 error, and remember now that this is concluding that there is no effect when in fact there is, and that's designated as beta, or oftentimes you'll see it referred to as statistical power, which is 1 minus beta. And the reason for this is just higher statistical power means that you will have a greater chance of detecting an effect if in fact it is there. And I'll talk about that more on the next slide. But there's a number of things that influence the type 2 error rate, or beta. One is the alpha level that you choose. As alpha decreases, the type 2 error rate increases. As sample size gets larger, the chance of making a type 2 error goes down because you have a better, often will have a smaller standard error. As the effect size gets larger, the numerator term, say in our t-tests, the chances of making a type 2 error go down. And whether you use a one-tailed or a two-tailed test also influences this. In some cases, we a priori say, well, we expect that this new diet is going to result in greater growth rates. So we will use a one-tailed test to test this hypothesis. And in that case, you're limiting the outcomes that you're considering, and that will increase the power of your test or decrease beta. What's important to consider here, however, is if in fact the outcome is the opposite of what you predicted, in other words, they grow slower, then really, if you're being honest, you shouldn't even conduct a statistical test. You conclude immediately that the outcome is not what you expected. Usually we'll do two-tailed tests. We'll say we're not sure if it's going to be an increase or a decrease, and we'll consider a difference in either direction to be something of interest. Term that you'll often see in the literature is what's referred to as statistical power, 1 minus beta. So essentially it's detecting an effect or difference when in fact there is an effect. Now it's really important to calculate power before you conduct a study. That's really the time when this concept is most appropriate. And you can either use a pilot study to try to estimate your effect size and experimental error, or you sometimes can go into the literature and say, well, here's a similar kind of situation where these folks were looking at the response variable that we're interested in. This is the difference they found. This is the experimental error. And then using that and the alpha level that you set, you can calculate your power ahead of time and say, well, we thought we were going to only need 20 plots, but we actually need 40 plots in order to detect, have a good chance, a 90% chance, say, of detecting a difference if in fact it exists. Now, one thing that you might see in the literature, especially some of the older literature, is to calculate power of a test after the fact. So you go out and you do a test of some treatment effect, maybe fertilizing on growth of grasses or whatever, and you fail to detect an effect. You think, well, was my failure to detect an effect due to the fact that I just had low statistical power, or was it due to the fact that there really is no difference, that I had a good chance of detecting it, but I just didn't do it? Well, it turns out that's inappropriate, because the power that you calculate is going to be completely determined by the specific alpha level that you found when you did your first test. And you can take a look at Honig and Heasy 2001 to get some more background on this. So this brings us back to this whole question of level of significance, or alpha level, that is chosen. The question is, should you set an alpha level? You often see that, as I said, in the literature, set at 0.05. In my opinion, there's really no need to do that anymore. Most computer programs will give you an exact p-value, and it's more appropriate to present that p-value and let the reader interpret it versus saying, well, I'm only going to conclude there's a difference if the p-value is below 0.05 or 0.001 or whatever it might be. That way, if it's close to 0.05, for instance, a reader can look at that and say, well, that's good enough for me. I think that's pretty significant, and I'm going to go ahead and implement a new policy based on that. So, for example, the question is, what does a p-value of 0.056 mean? Do you just cut it off and say, well, that's greater than 0.05, so nothing going on here? Or do you think, well, there's, that's pretty low probability, so perhaps this could be something of significance? So now let's move on to confounding or lurking variables. This is a topic that's covered in Joyner's paper. He doesn't use wildlife examples, so I will let you read that and get the basic concepts through him, and we'll go through some examples that might be more applicable to wildlife situation. So Joyner defined a confounding or lurking variable as a variable that has an important effect and yet is not included among the predictor or independent variables under consideration. In most cases, it's not a variable of interest, but if you're not accounting for it, it may affect your ability to detect treatments. And this can happen both in experiments, as we'll talk about, and especially in correlative studies. It can be omitted from the analysis either because its existence is unknown, its influence is thought to be negligible, or the data simply aren't available. And in many cases, it's just considered a nuisance parameter. Now, obviously, the problem with lurking variables is they're hard to identify. If you don't know that a variable might be affecting your results, you may not even think of looking at it. But there are some ways that statisticians have to examine the data and see if there might be some effects going on that we weren't aware of. And again, Joyner is a good place to get kind of the nuts and bolts of this. But just to review some of the things that can be affecting this and how you might detect them. Time order. So if you have a scale that's losing power over time, and the results that it's given you might be affected by the strength of the battery. You can order your measurements over time, and sometimes that change will become very obvious in what we call the residuals of your relationship. Um, a day of week could have an effect if you're doing an experiment in the laboratory and the temperature in the room is set for different temperatures and different days of the week, or between the weekend and weekdays and so on. Spatial arrangement of your samples. Again, if you have a laboratory experiment, if samples are close to the wall and it tends to be cooler close to the wall, that could have an effect. If the light varies across the room and your experimental units are arranged in different positions relative to that light, lots of different effects of spatial arrangement. And again, you might pull out those samples that were back along the wall and compare them to those that are further away and see if you see any sort of systematic change in your residuals. The residuals are just defined as the difference between the mean for a particular group and the value for that particular experimental unit. And you might use some other diagnoser variable. Something common in wildlife would be individual collecting the data. We know that there's a lot of individual variation in studies that we do with respect to simple things, even like measuring the length of a bird wing, or the weight of a fish, or percent cover of grass in a plot, and so on. So pulling out who collected the data and seeing if there's systematic variation from person to person can be important. And this is really where your knowledge of the biology and ecology of the system can be important, and you need to consider what lurking variables could be out there and actually try to test for them to see if there might be an effect going on. So you can probably all think of some examples from studies that you've been involved with, and in fact, that's one of the assignments for this week. I'll just go over a couple of other examples that have come up in studies that I've been involved with. Some equipment can be affected by temperature. As I mentioned, cold batteries can sometimes affect the readings that you get on an electronic scale. The same is true of what we call Pasola or spring scales. Colder temperatures will make that spring tighter and might give you biased readings and so on. In bird point counts, an area that I've been involved with for many years, we know that there's many nuisance variables that can affect how many birds you count at a particular point. So in this process, you go and stand at a point for 5 minutes or 10 minutes, whatever it might be, count all the birds that you can see or hear around that point, and use that to estimate their abundance. But we know there's many things that can affect your detectability of birds around that point. Detection rates decline from morning to afternoon as birds stop singing. Temperature can have an impact on movement rates and call rates of birds, and of course season, depending not only whether you're in the breeding season, migration, or wintering season, but even within a season, we know that there's can be big changes in the rate at which birds sing, for instance, from early in the season when they arrive on their territories and sing a lot to later in the breeding season when their singing rates drop down. If you're not taking those into account or somehow trying to correct for them, you can see how that could have either just negate any strong effects that you might see due to the comparisons you're making or the treatments you're looking at, or in fact, in some cases, give you completely erroneous results. Time can have effects in many different ways. For example, it's been found that the phase of the moon can influence the number of captures of small mammals. So when the moon is out and bright, and the animals presumably are more likely to be seen by predators, their activity drops down, so you catch fewer. There's ways of correcting for that with mark recapture techniques, but if you're just using capture rates without adjustment, that could lead to some biased numbers. Another example in radio telemetry, we know that many individual animals have certain patterns of movement that they might exhibit throughout the day, start near their night roost and then move to some feeding area in the morning and then move to a mid-morning roost and so on. And so you want to be careful not to monitor if you're trying to get information, say, on the home range of common ravens like we were in Redwood National Park. You don't want to go and monitor raven number 1 first thing in the morning, every morning, and then go to raven number 2 a little bit later in the morning, because you may be tapping into some daily pattern that those birds are involved in. And so you want to randomize the order in which you visit those individual birds so that you spread your observations out over the course of the day and presumably get a better estimate of their home range.
Now I want to go through an example of 2 studies that I've been involved with to talk about how a new variable that we identified could potentially have greatly influenced our results. So we were interested in what factors influence annual survival of this bird, the Swainson's thrush. Breeds in fairly wet forests in the western United States, up across the Taiga and over into the eastern U.S., but we were focusing primarily on the west. And we knew that there were a variety of factors that could influence annual survival in songbirds. A lot of studies have been done on this. Our main interest in this study was looking at the effects of climate cycles on bird survival. But we knew in order to detect what might be a fairly subtle effect of climate cycles, and I'll talk about that, we needed to take into account other factors that might influence Swainson's thrush survival. We knew that age has a big influence, so we restricted our analysis to adults only. We knew that the region that a bird was captured, and this is all based on mark recapture data where we catch birds in mist nets at a variety of different locations across the West, release them, catch them again over time. We'd go back to those same locations year after year. And so we divided our capture site locations up into these different regions to see if perhaps possibly region had effect. We also simply looked at latitude overall, thinking that maybe just a continuous variable of latitude might be important. We examined what's known as resident versus transient effects. So whether a bird had been captured multiple times at a site, indicating that that bird was a breeder and a resident at that site, versus birds that were only caught once the first year that they were banded, indicating that they might be migrants or floaters. And then there were 3 climate cycles that we were interested in. One was the El Nino Southern Oscillation, strong pattern that has a warming effect on the East Central Pacific near South America during El Nino years, and cooling during La Nina years. There's also a pattern called the North Atlantic Oscillation, which obviously is over in the Atlantic Ocean, that generally has more of an influence on precipitation and temperature patterns in the eastern United States, but has been shown to have potentially some effects in the West as well. And then finally the Pacific Decadal Oscillation, which is a much longer term change in temperature patterns in the northern Pacific that, again, has been shown to have some fairly strong effects on precipitation temperature patterns across the western U.S. So when we ran our analysis, including all of these various variables I went over, we did find that the El Nino Southern Oscillation pattern had a very strong effect on annual survival of Swainson's thrushes. And in fact, as the El Nino index increased, survival increased. And we think this has to do with rainfall along their northern migration route. We also did find that region was important. So here we've plotted survival as a function of the El Nino variable for region 7 up north versus region 1 down south. And the main thing that you see different here is if you look at the the x or the y-intercept, you see that in general survival down in region 1 tends to be lower for the same level of the El Nino index than it is in the northern region. Residency was also a very big effect. So obviously residents had higher survival than the transients. And we found what's known as a region by residency interaction. In other words, the influence of residency on survival changed from region to region. So again, that's not something that's biologically necessarily very interesting, but by including that effect, we were able to get a better test of our influence of our factor of interest, which was El Nino. A few years later, I got a call from a postdoc at UCLA, and he had information on the intensity of West Nile virus in locations across the entire continental United States. And this was the first time that we had good large-scale information on West Nile virus intensity across many, many years. And immediately we became interested in knowing, did West Nile virus have an influence on survival of Swainson's thrushes? Now, laboratory experiments and other work had indicated that it probably didn't have a big effect on Swainson's thrush survival, but no one had really looked at it in wild birds before. And so when these data became available, we were excited, but we were also worried that perhaps the ENSO effect that we had detected from our earlier analysis really wasn't due to the El Nino Southern Oscillation, but perhaps because survival had been influenced by this other variable, this lurking variable, West Nile virus. And by not taking that into account, we had come to the wrong conclusion. So here I've plotted Swainson's thrush survival over time relative to when West Nile arrived, and most of our data were from the western U.S., so that's when the arrival date was identified. And what we found is that there was evidence of effect of West Nile virus on survival after it arrived in 2002.
So what was the upshot of the new analysis? Well, when we added West Nile virus, we actually found that the ENSO effect was larger with West Nile virus intensity included in the analysis versus without. You can see that the beta, or the coefficient for ENSO, goes from 0.101 to 0.112, so indicating that there's a stronger effect of ENSO on survival. The standard errors essentially didn't change, stayed at 0.019 versus 0.020. So we didn't reduce the variance associated with the ENSO effect, but the effect itself was found to be stronger, probably because in those later years when West Nile was having an impact on survival, there was also some effects of ENSO that were masked. So in this case, we can breathe a sigh of relief and go, okay, here is a lurking variable that we hadn't considered. When we put it in, we still find that the effect is strong, in fact, even stronger. But you could see if the West Nile intensity was strongly correlated, for instance, with ENSO, it could have completely wiped out the ENSO effect. And in this case, it's a little bit harder to imagine because West Nile only arrived in that area later in the period, but you know, in other kinds of variables that had been present throughout, you could see how that might have a big effect. So just again, a caution, always think about what other factors might be influencing your variable of interest or your dependent variable, and make sure you're taking into account as much as you can.